Hello and welcome to the video on Genetics Simplified Key Concepts and Problem Solving for CSIR NET Life Science. There are many genetics key concepts you study in Unit 8, that is Inheritance Biology of CSIR NET Life Science exam, like Mendelian principles, Mendelian ratios, probability rules, extensions of Mendelian principles, gene mapping methods, microbial genetics, pedigree analysis, mutation, structural and numerical alterations of chromosomes, and many more. Knowledge of these key concepts are important from exam point of view, as questions are frequently asked from these concepts. So here are few concepts which you must understand and learn for CSIR NET Life Science exam. The very first concept you're going to learn in this video is about genetic mapping and physical mapping. To determine the relative or exact distance and location of genes on chromosomes, mapping is done. There are two types of mapping, genetic mapping and physical mapping. So let's understand the difference between them. Genetic mapping is the process of determining the order of and relative distance between genetic markers on a chromosome based on their pattern of inheritance. That is how the genes are transmitted from parent to offspring. So genetic mapping is used to find out the relative position of genes and order of genes in a chromosome. So as we all know that chromosome has a centromere region and a short arm, that is P arm, a long arm, that is Q arm, telomere region is there, subtelomeric region is there, and genes are located on chromosomes. So let's say we have the three genes A, B and C. So we want to determine whether the order of these three genes is A, B or C, or is it C, A, B, or is it B, C, A, and so on. So we want to determine the order of genes as well as we want to determine the relative position of genes, the location of genes on a chromosome. So for that we use genetic markers, which are the genes or can be specific DNA sequences that are inherited along with genes of interest. Physical mapping, however, is the technique which is also used to determine the order, used to find the order, but it is used to find the physical distance, not the relative distance between DNA base pairs by DNA markers. So physical mapping, it provides the accurate physical distance between the DNA markers in terms of nucleotides. So DNA marker can be a restriction recognition site. So as you can see here that the distance between the genes, they are determined in terms of base pairs. Now, how can we construct genetic map or a physical map? The primary basis for constructing a genetic map is the pattern of inheritance observed among the markers. It uses recombination and linkage studies to find out the relative distance and inheritance pattern. Scientists, they have observed that the genes which are closely located in a chromosome, they have a less chances to undergo recombination. Like if we compare A and B and A and C, you will find that A and B are more closely located than A and C. And therefore, they have less chances to undergo recombination. A and B have less chances to undergo recombination. So they are rarely shuffled by recombination, and therefore the scientists, they can conclude that as the genes, they rarely shuffle by recombination, they must lie close together on the same chromosome, and more likely they are to be inherited together. We call such genes as linked genes, which are always inherited together, whereas the loosely linked genes, they are more frequently shuffled by recombination and therefore they lie far apart. So therefore, genetic maps uses recombination and linkage studies to find out the relative distance and inheritance pattern. Now, unlike genetic maps that rely on inheritance pattern, physical maps, they directly measure the physical distances between the markers. So physical mapping, they use molecular techniques to identify specific DNA markers or sequences along the chromosome. So usually physical maps, they can be constructed using various techniques such as DNA sequencing, restriction mapping, and hybridization techniques like fluorescent in situ hybridization mapping. So these methods, they provide precise information about the physical distances between DNA base pairs. Now, distances on genetic maps, they are measured in map units. In short, we write MU or centimorgan. So as you can see here, the distance between the genes, they are measured in map units or centimorgans, which represents the likelihood of recombination events occurring between markers during meiosis. And kindly note that one map unit is equal to 1% recombination occurring between two markers. So genetic maps determine the probability of recombination events between 2 points. Physical map, it measures the distances in base pairs or kilobases, where 1 kilobase is equals to 1000 base pair. So as you can see here, the distance is measured in base pair. And physical maps, they determine the number of bases between 2 points. Like if we are considering these 2 points, then we can determine the number of bases which are present between these 2 points or genes. And if you compare genetic mapping and physical mapping, so genetic mapping is less accurate in comparison to physical mapping, as here we are determining the relative distance and not the physical distance. And genetic mapping, they provide firm evidence on genetic disorders associated with one gene like cystic fibrosis and muscular dystrophy, or 2 genes, example diabetes or cancer. Whereas physical mapping, they are important to identify the origin of diseases, whether they are inherited or arise due to a random mutation. So let's have a look at the type of question which can be asked from this topic in CSIR NET Life Science exam. So here is one part C question. A figure is given which depicts the relationship between a genetic map for 4 genes, A, B, C, and D, and their corresponding physical map. So as you can see here, this is the linkage map. So genetic map is also called as linkage map, and the physical map. And following statements are made to explain this relationship. Statement A is more number of recombination events occur between A and B as compared to B and C. On the other hand, B statement is lesser number of recombination events occur between C and D as compared to B and C. And then we have statements C and D. So they are contradictory. That according to statement C, the physical distance between A and B is less than that between C and D, and region between A and B is more recombinogenic. Whereas statement D is that A and B, the physical distance between A and B is less than between C and D, and thus the region between A and B is less recombinogenic. So here you can see they have just changed. In this question, only one word is changed in these 2 statements. So you have to read the statements carefully. And then statement E and F are again contradictory. Statement E is physical distance between C and D is more than that between B and C. The region between C and D is less recombinogenic. Whereas according to statement F, the region between C and D is more recombinogenic. So you have to choose the statement which are correct. So although this question appeared to be lengthy, but such type of questions are very easy. You just need to learn to analyze the figure given, the map given, and based on that you have to go through the statements and find out whether they are correct, and maybe in the exam incorrect statement can also be asked. So what is the strategy of solving such questions? The very first thing is you have to read the question carefully, go through the figure given carefully, understand it, and then go through each statement one by one and find out which statements are correct as correct statement is asked in the exam. So you have to pay attention to this word, that whether correct statements are asked or incorrect statements are asked. They can even in the exam, you may also ask to find out 2 correct, 1 incorrect statement or vice versa, right? So answer the question accordingly. So as we have studied that, recall here the concept of genetic map and physical map. As we all know that genetic map is used to find out the gene distance, relative distance, whereas physical distance is determined by the physical map. So physical distance is the actual distance between 2 points. So that is why physical distance are preferred in comparison to genetic map distance. And as we have studied that the farther the genes on the chromosome, there are more chances of recombination, right? So as you can see here, the distance between gene A and B on linkage map is 20 centimorgan, B and C is 10 centimorgan, and C and D is 10 centimorgan. Whereas if you see the physical map, the distance between A to B, B to C and C to D, so you can see A to B is 20 MB and B to C is also 20 MB, but C to D is 40 MB. So physical distance between A and B and C is less in comparison to C and D, right? So now we go through each statement one by one. Statement A is more number of recombination events can occur between A and B as compared to B and C. So recombination events as we are talking about, so we'll look at the linkage map because they are based on the recombination and linkage studies. So A and B distance is 20 centimorgan and B and C is 10 centimorgan. So as we all know that farther the gene apart, there are more chances, like more the distance between the genes, more chances of them to undergo recombination. So yes, this is correct. A statement is correct. Then if we see B statement, lesser number of recombination events can occur between C and D as compared to B and C. So when we look at the linkage map, B and C distance is 10 centimorgan and also C and D is 10 centimorgan. So this is not correct statement because we cannot infer by looking at the linkage map, because distance between the genes B and C and D is 10 centimorgan, the distance is same. So you cannot infer that lesser number of recombination events can occur between C and D as compared to B and C. Now as we know that C and D are contradictory statement, only one word is changed over here in these statements. So the statement is that physical distance, now we have to look at the physical map. Physical distance between A and B is less than that between C and D. So yes, this is correct. Here A and B distance, physical distance is 20 MB, whereas C and D is 40 MB, right? So the region between A and B is more recombinogenic. No, this is incorrect. It will be less recombinogenic, right? Because chances of recombination will be more in C and D in comparison to A and B, and even if you compare B and C. So therefore, statement D is correct and C is incorrect. So when I say D is correct, so it is given only in option 3. And we don't have the combination where A and D both are given together, right? So we'll confirm the answer. Let's have a look at the F statement also, E and F statement also. So when I say, so if you look at the statement E and F, again there is a change in single word, that physical distance between C and D is more than between B and C. So region between C and D is less recombinogenic or more recombinogenic? It will be more recombinogenic. And therefore statement D and F both are correct, which is given in option 3, and therefore it is the answer. Another important concept is pedigree analysis. In CSIR NET Life Science exam, you may get direct question on pedigree analysis, where a family tree is given and based on its analysis, you have to determine the inheritance pattern of a disease. Or sometimes a pedigree analysis questions are asked in combination with probability rules, where you have to determine the probability of a particular genotype in a progeny, or you have to determine the probability of a genotype in a parent. So let's understand about pedigree analysis in detail. Pedigree is a pictorial representation of a family history, essentially a family tree that outlines the inheritance of one or more characteristics. So let's understand the inheritance pattern of autosomal dominant trait and X-linked dominant trait through pedigree analysis. Autosomal dominant traits are the traits which occurs due to the presence of autosomal dominant allele, that is, presence of just one copy of the dominant allele is sufficient to express the trait. Whereas X-linked dominant inheritance refers to genetic conditions associated with mutations in genes on X chromosome. So a single copy of the mutation is enough to cause the disease in both males who have only one X chromosome and the female which has two X chromosome. So because it's a dominant trait, so a single copy of mutation is enough to cause the disease in both males and females. So let's do the comparative study. Autosomal dominant traits, they appear in both sexes with equal frequency, and both sexes are capable of transmitting these traits to their offspring. So as we all know that every person with a dominant trait must have inherited the dominant allele from at least one parent. So therefore, affected offspring, it must have an affected parent unless they possess a new mutation. So as you can see here, this is an autosomal dominant trait which occurs equally in males and female. So here you can see that the square represents the male, the circle represents the female, and this is a couple as it is connected with a line. And then, so this is the first generation, the second generation, that is the progeny of this couple. So you can see that male and female, so where you can see the female child is affected and male child is affected, whereas this female child, the third and the fourth one, the male child, is not affected. So if the progeny, the offspring is affected, that means they must have an affected parent because they must have inherited the dominant allele from at least one parent. And therefore, autosomal dominant traits, they do not skip generations. Exception to this rule arise when people acquire the trait as a result of a new mutation or when the trait has reduced penetrance. On the other hand, X-linked dominant traits, they appear in male and female, both male and female are affected, although they often affect more females than males. Because as we all know that males have one X chromosome only, and where this X comes from the mother and Y comes from the father in males. So therefore a male will inherit a X-linked dominant trait only from his mother. So affected sons must have an affected mother, and affected daughters must have either an affected mother or an affected father, because as we know in females there are two X chromosomes, so they can inherit the trait either from the affected mother or an affected father, because one of the X chromosome comes from mother and one of the X chromosome comes from father in a female. So since father is also contributing an X chromosome, so affected fathers will pass the trait on to all their daughters, not to their son, because if it is a son, then Y chromosome will come from the father. So that is why the trait is not passed from father to son. Affected sons, they must have an affected mother. So as shown in this pedigree, affected males pass the trait on to all their daughters and none of their sons. So as you can see here, this is the affected male and this is the unaffected female. So this is a couple and they have the progeny. So the offsprings are 2, 3, 4 and 5. So you can see all the daughters are affected. And second, which is a son, is not affected. So these daughters, they have inherited the X chromosome from the father who is affected. So X-linked trait they have received from the father for the disease. And X-linked dominant traits also do not skip generations. Now, for autosomal dominant trait, when one parent is affected, that is heterozygous, and other parent is unaffected, the chances of their offspring to be affected is half. So let's say A is the affected allele. So if one parent is affected and is heterozygous, so that means this is the genotype of the parent, and another parent is unaffected. So since it's an autosomal dominant trait, so if a parent is unaffected, it means it must be having a recessive allele in homozygous condition. If a cross occurs between these two individuals, so let me draw the Punnett square. We can write down the gametes formed by one parent on one side and another parent on another side. So this is parent 1, this is parent 2. So these are the gametes of parent 1, this is of parent 2. So this will be the genotype in the progeny. So out of 4 possible combinations, you can see 2 are heterozygous and 2 are homozygous recessive. And since the disease is associated with a dominant allele, so probability of offspring being affected is 2 out of 4, or I can say half. Similarly, the probability on offspring being unaffected is also 2 by 4 or half. On the other hand, if both parents have the traits and are heterozygous, so now let's consider the condition when both the parents are heterozygous, then each parent will produce two types of gametes, one with the dominant allele and another with the recessive allele. So Punnett square will show 4 possible combinations: homozygous dominant, which will be affected, heterozygous, which will be affected, and homozygous recessive, which will be unaffected. So therefore, the probability of an offspring for being affected, as will be in this case in homozygous dominant or heterozygous, so the probability is 3 out of 4. X-linked dominant trait affected mothers, if heterozygous, will pass the trait on to half of their sons and half of their daughters, as shown in second generation and fifth female, which is married to a normal male. So they have offspring 6 to 11. So here you can see that the son 9 and 11 are affected, whereas 7 is unaffected, and the female child 6 is affected, whereas 8 and 11 is not affected. So basically, as we all know, the trait is carried on the X chromosome, and only one copy of the dominant allele from either parent is necessary for individual to express the trait. So if the mother is heterozygous, so let's say XA is the X chromosome with the dominant allele for the trait, and this is the another X chromosome with the normal allele. And let's say the father's genotype is having the normal allele on X chromosome. So the inheritance can be mapped out as follows. So we'll write down the genotype of the gametes formed by the male and female. So here you can see that the son will receive the Y chromosome from the father and one X chromosome from the mother. It can be the X chromosome with the affected allele or with the normal allele. So that means there's a 50% chance that a son will inherit the affected allele, so it will be affected, and 50% chance that it will inherit this genotype and will be unaffected. However, in case of the daughters, the daughter will receive one X chromosome from their father and one X chromosome from their mother. Now, father will always pass the normal allele, as father is unaffected, whereas mother can pass either the X chromosome with affected allele or with the normal allele. So that means again there will be a 50% chance the daughter will be affected and 50% that it will be unaffected. So one trait which is usually considered to be autosomal dominant is familial hypercholesterolemia, an inherited disease in which blood cholesterol is greatly elevated owing to a defect in cholesterol transport. Whereas an example of an X-linked dominant trait in humans is hypophosphatemia, also called as familial vitamin D resistant rickets. Next is autosomal recessive trait and X-linked recessive trait. Autosomal recessive inheritance is a genetic condition that can occur when the child inherits one copy of a mutated gene from each parent. So for example, let's say this is the normal allele and this is the mutated. So when the parents are heterozygous and each parent will contribute the recessive allele, then only the individual will be affected. So autosomal recessive traits normally appear with equal frequency in both the sexes, and as I mentioned, the traits appear only when a person inherits two alleles for the trait, one from each parent. And if the trait is uncommon, both parents carrying the alleles are heterozygous and are unaffected. Consequently, the trait appears to skip generation. So as you can see here, the parents are unaffected, but the child, the offspring, as you can see in second generation, is affected. So this is only possible when each parent have contributed the recessive allele, as the recessive trait always appear in homozygous recessive condition. However, X-linked recessive disorders, they are also caused by variants in genes on X chromosome. Now, as we all know that males have only one X chromosome and the females have two X chromosome. So which means that one altered copy of a gene in each cell is sufficient to cause the condition in males. However, in females who have two X chromosomes, a variant would have to occur in both copies of the genes to cause the disorder. And this is the reason that the trait appear more frequently in males, because the males inherit only a single copy of allele to display the trait. And females, as mentioned earlier, that they must inherit two copies of the allele, one from each parent, to be affected. So let's say this is the X chromosome with altered copy of the gene. And also, as a male inherits X chromosome from his mother, affected males are usually born to unaffected mothers who carry an allele for the trait. So as you can see here, this is the unaffected female carrier. So as we know, a female has two X chromosomes, so that means it will have one X chromosome which is with the mutated gene. So itself the female will be unaffected, because in case of females, a variant would have to occur in both copies of gene to cause the disorder. But you can see that the offspring 3 is affected, as males have one X chromosome, and if it receives the affected copy from the mother, then it will be affected. And because the trait is passed from unaffected female to affected male and then to unaffected female, it tends to skip generation. So if you see the third generation, the 7th offspring, it is a carrier and it will be unaffected, because it must have the one X chromosome with the normal, which is normal, and a normal gene, and another with the affected. Then another important characteristic of X-linked recessive trait is that they are not passed from father to son, because a son inherits his father's Y chromosome and not his X. So as you can see in this figure, no case of a father and son who are both affected. However, all daughters of affected fathers are carrier if their mother is homozygous for the normal allele. So for autosomal recessive trait, affected offsprings are usually born to unaffected parent, as you can see here. Both the parents in first generation, if you see, both the parent 1 and 2 is unaffected, but the offspring 3rd is affected. However, for X-linked recessive trait, affected sons are usually born to unaffected mothers, as we can see in the first generation and the second parent. Then for autosomal recessive trait, when both parents are heterozygous, the chances of offspring to affect it is 1 by 4. So let me show you the cross here. So let's say for autosomal recessive trait, if both parents are unaffected, so that means they are heterozygous. So this is parent 1, this is parent 2, and let's write down the gametes formed by each parent. So in the progeny, we can have homozygous dominant condition, heterozygous condition, and homozygous recessive condition. So as we know that when both parents are heterozygous, only 1 out of 4 of the offsprings are expected to express the trait. However, for X-linked recessive trait, if mother is carrier, that is heterozygous, the chance of her sons to be affected is half. So let's consider the cross. So let's say mother is a carrier. So this is the normal allele and this is altered. So female is heterozygous, mother is heterozygous, and let's say we have an unaffected father. So in such case, the progeny will have the genotype. So you can see daughters, if we see the daughters, so this will be unaffected, 50% will be unaffected. This will also be unaffected, but will be the carrier. And in case of son, so this will be unaffected and this one will be affected. So 50% unaffected, 50% affected. Also kindly note that autosomal recessive trait is more likely to appear in pedigree where two people within the same family meet, because there is a greater chance of both parents carrying the same recessive allele. So mating between closely related people is called as consanguinity. So as shown in the pedigree, so third and fourth are the first cousins. So if both are heterozygous for the recessive allele, so when they meet, 1/4 of their children, as shown in the cross earlier, are expected to have the recessive trait. So if the parents have this heterozygous condition, 1/4 of their children will have are expected to have the recessive trait. And a number of human metabolic diseases are inherited as autosomal recessive trait. One of them is Tay-Sachs disease, whereas an example of X-linked recessive trait in human is hemophilia. Now let's have a look at the type of questions which can be asked based on pedigree analysis. A pedigree of an inheritable disease is shown below. So based on pedigree analysis, we have to find out what type of inheritance does the disease follow. Autosomal dominant, X-linked, X-linked recessive, or autosomal recessive. So as we know that autosomal dominant, for autosomal dominant disease, one copy of the allele is sufficient to cause the disorder. Whereas for autosomal recessive, the two affected alleles in homozygous condition will be needed to cause the disease. So if we analyze the given pedigree and if we write down the genotypes, so like here you can see this is the first generation, this is the second, and this is the third. And as denoted that this is a carrier female. So let's number it as 1, and this is the unaffected male. So to answer this question, you have to analyze the pedigree given family tree carefully. And the details they have mentioned that unaffected female is represented with a circle, carrier female with a circle with a dashed line, and affected female with a circle which is shadowed. And unaffected male is represented with a square, carrier male with a square with a dashed line, and affected male with a square which is shadowed. So as we know that males, they have a genotype XY and female have 2 X chromosome. So let's suppose if one of the X chromosome, like the point is whether it's autosomal dominant or X-linked dominant or X-linked recessive or autosomal recessive. So from the pedigree analysis, one thing is clear that it is not an autosomal dominant or recessive disease. It could be X-linked dominant or X-linked recessive. So if we consider, let's say this is 2 X chromosome. So since the male is unaffected, so it will definitely have a normal X chromosome. Whereas female, it's a carrier, so it is heterozygous. So let's denote one of the X chromosome with the affected allele. So let's say C is the affected allele on the X chromosome. Now in the second generation, you can see that let's number it as 1, 2, 3, 4, 5, and 6. So we have the first one is the unaffected male, so XY. Second is a carrier female, so again it will be heterozygous. Then third is an unaffected male. Fourth is an unaffected female. Then fifth is an affected male. So it has inherited the affected X chromosome from the mother, and 6th is unaffected female. Then if we look at the 3rd generation, so again we have, let's number them. So first is an unaffected female, second is an unaffected male, then we have a carrier female, then we have an unaffected male, then carrier female again, unaffected male, then carrier female again, and then we have the unaffected male. So on analyzing the pedigree, you can see that in the 1st generation, the female parent is the carrier and is unaffected. Then in 2nd generation, you can see that the 5th child, the 5th offspring is affected, but none of its progeny, that is from 5th to 8th, is affected. It's only that if you see in the 3rd generation, the 5th child and the 7th child, which is a female, the daughters, are the carriers, but they are not affected. So therefore, disease we can say is X-linked recessive and not X-linked dominant. The next concept is mutation. So as we all know that mutation can occur spontaneously in nature, or it can be induced by the factors like X-ray or UV radiation. So we can classify the mutation based on the inducer, whether it occurs spontaneously in nature or is it induced. Then even we can classify it on basis of the cell type in which the mutation is occurring, whether it is a somatic cell. So if the mutation occurs in somatic cell, it is non-heritable, but if it occurs in germinal cell, then yes, definitely it is heritable. Then whether the mutation occur in forward direction or reverse direction, so this is another way of classifying the mutation. Then whether the mutation is occurring at the chromosomal level or occurring in a gene, or is it a point mutation. So if it is occurring in a chromosome, so whether there is a structural change or whether there is a change in chromosome number. Structure change can happen because of deletion of a region or inversion of a region or duplication or translocation. Similarly, the changes in chromosome number can occur. So again, we have further classified the mutation. And if it is occurring in a gene, so whether it's a deletion in a gene or it is an insertion in a gene, or is it a missense mutation, silent mutation, or substitution. So you can see that this is a flow chart which has been specifically made by Biotechnica for quick revision and recall of this concept. So this concept is covered in detail in the regular classes. So this is all for now. Thank you and happy learning.